Well, so we start Unit 2, which arguably is the, I don't want to scare anybody, but just letting you know, it's arguably the hardest one in the course. Genetics and metabolism took pretty abstract concepts. Unfortunately, there is no way I can, I try to make them more relatable, but they're still pretty abstract. Okay? So, follow me, ask me if you have any questions, you know. If you don't understand something, I want everything to be crystal clear. First thing first, I want to remind you about the structure, the DNA, basic stuff. DNA consists of building blocks called nucleotides, and this picture in the corner shows you a typical nucleotide. In the nucleotide, you can see three parts. Highlighted by beige color is the nitrogenous base. It's called nitrogenous because it's heterocyclic nitrogenous compound. Okay, there are different types of nitrogenous bases. There are pyrimidines, like this two, cytosine and thymine, and there are purines, like this two, adenine and guanine. So in the DNA, two pyrimidines are C and T, and two purines are A and G. You need to know which nitrogenous bases are purines and which are pyrimidines. I do not ask any questions involving chemical structures. You do not need to know the structures themselves. Hooray. Okay? Yes, I'm with you on that. But if I ask you, you know, adenine is purine. Now, when nitrogenous base, here in the corner, combines with a sugar, so-called deoxyribose sugar, they form a structure known as nucleoside. The sugar is called deoxyribose because here, at the 2' carbon, there is no hydroxyl group, just hydrogen. So deoxy, no oxygen. Does that make sense? Now this seemingly small detail, the absence of OH group at the 2' carbon, makes DNA very chemically stable. Okay? Which is, of course, a necessary feature for the DNA because it's going to store genetic information and we want it to be incredibly stable. It also gives us a lot of opportunities to peek into the past. For instance, now we have an entire genome of a woolly mammoth, and I totally think we got to clone it and bring it back. And as soon as we recover, and there is a work on recovering the genome of dinosaurs from amber. There are fragments of dinosaur DNA captured in amber. Totally think if we get the entire genome of T-Rex, we should totally bring it back too, because what can go wrong? Everybody knows who watched Jurassic Park. Oh no, it would be. It would be fine. Oh, it would be totally fine. They would have to live in some, you know, pretty warm place like Florida, but come on, T-Rex in Florida. It's like yesterday's news. So this part is nucleoside. Just terminologically, you know, nucleoside is the base plus sugar. When you throw in phosphate, Okay? Nucleoside plus phosphate becomes a nucleotide. Now you may ask, wait a minute. Why do we have so many different terms? Turns out, if you can prepare a drug that looks like nucleoside, it can work as an inhibitor of various cellular processes, such as DNA synthesis or RNA synthesis. And these drugs, so-called nucleoside analogs, are used to treat diseases like herpes or HIV. So, to kind of wrap it up, every nucleotide contains nitrogenous base, sugar, and a phosphate. Nitrogenous base and sugar form nucleoside. Okay? Now, what are the roles of these three parts of a nucleotide in forming the structure of DNA? Phosphate and the sugar form what is called a backbone of the DNA molecule. Now, if you will look at the sugar residue right here, you will see that all the carbons of the sugar of the deoxyribose are numbered from 1' to 5'. If you're curious why it has to be with a prime, because numbers without prime are used for numbering atoms of the nitrogenous base, but it's not really important for our conversation. Just accept the numbering from 1' to 5'. Can you see the numbers right there? Now, you may notice that phosphate residue, this one, it links 3' carbon of one sugar and 5' carbon of another. And the phosphate residue here also links 3' carbon of one sugar and 5' carbon of another. Does that make sense to you? So all phosphates will connect 3' carbon of one deoxyribose and 5' carbon of another. Which means that these sugars are oriented in a certain way, kind of a head to toe. Does that make sense? And that gives that molecule, the DNA molecule, directionality. Okay? You understand what I'm saying? The best analogy that I came up with about this is, people, like a lot of people dancing conga. When you dance something like that, you have to stand in a certain order, right? So each person faces the back of the person in front of them. If people will randomly position themselves, then this giant chain of people will not be able to move, correct? Because they will be like all moving in different directions. So that precise, exact orientation of 3 to 5', right here, 3 to 5', means that DNA molecule is directional, and it means that it can be read in a certain direction. It turns out DNA is always read from 3' to 5'. Like we're reading from left to right. Makes sense, correct? And it doesn't have to be left to right. If you're reading Arabic or Hebrew, it's going to be right to left. If reading Old Chinese, it's going to be top to bottom. But there is a directionality in every language, and this is the language. Am I clear? So first of all, DNA is directional. Now, what is the organization of the entire molecule? DNA is double helix. I told you before, if I want you to memorize the number, I will tell you to memorize the number. Since I don't tell you about this number, you don't have to remember any numbers about the DNA molecule. But I do want you to know that it's a double helix. Now, what holds two strands of the DNA together? Hydrogen bonds between pairs of nitrogenous bases. It is a rule that A pairs with T, G pairs with C. Okay? Those are most energetically efficient pairs. Do I make sense? Do you understand what I'm saying here? Now, which pair, AT or GC? Well, I kind of give you an answer. AT pair forms weaker pair than GC. Why AT is weaker? Only 2 bonds. Yes, only 2 bonds. And you can say, okay, that's an interesting factoid. Why is that important? It turns out that weaker pairs will be found in the parts of DNA that are more amenable for unzipping. You understand what I'm trying to say? So if you have a lot of ATs, AT pairs, you can easily separate the strands. If you have a lot of GCs, they're going to stick together much better. Correct? We good? Not only strands are forming double helix, strands are also anti-parallel, which means that one strand, so say you look at the molecule of DNA on the right-hand side of the slide here, right? It's a schematic representation, so you see that the strand on the left has 5' end on top and 3' end on the bottom. Then the strand on the right will have 5' end on the bottom and 3' end on top. Okay, so they are anti-parallel. If we want to characterize the structure of the DNA molecule, we can say it's the anti-parallel double helix, in which individual strands are held together by hydrogen bonds. Are we clear? Actually, here's the model. Fairly poor model, but that's the best we have. Model of the DNA molecule. So you see nitrogenous bases. There's a really long, like blue, yellow, red, and green. Okay. You see what I presume is phosphate, the white ribbon, and these orange D's represent deoxyribose. Okay, that's more or less all right description of the DNA structure. It's not exactly what it's supposed to be, but good enough. Now, if you would look at question, Victoria. Say it one more time. So in the picture on the right, okay, picture on the right is schematic. Does that make sense? On this picture, artist put the 5' end of the left strand on top and 5' end of the right strand on the bottom. If we flip them, nothing's going to change. As long as the strands are anti-parallel, it's a correct depiction of the DNA. Does that make sense? Did I answer your question? Good. Now, if you would look at the picture on the left, here, a better representation of the DNA structure, you may notice the DNA molecule is a bit asymmetrical. And due to that asymmetry, the molecule has 2 grooves. The major groove and the minor groove. Now, these grooves aren't just fancy parts. They are actually sites for the protein binding. A lot of proteins have to bind to DNA for different reasons. Now, they will bind into those grooves. Does that make sense? Now, the last thing that I want to say about the DNA structure here is so-called Chargaff rule. So Chargaff rule formulates really simple. In the double-stranded DNA molecule, the total percentage of adenine, or A, equals the total percentage of thymine, T. And the total percentage of guanine, G, equals the total percentage of cytosine, C. Now, do you understand why? And if you don't, it's totally fine. Do you need an explanation why that is so? Okay. Don't ever hesitate to say, I have no idea what you're talking about. Explain it to me. Now, this is how it works. Give me a second. I'll do an explanation and I'll repeat. Okay? So, we're going to describe now a double-stranded DNA molecule. Okay? We're going to have two strands. Strand 1, I'm sorry, and strand 2. Does that make sense so far? Now, we're not going to draw each strand like completely. All I want to do is to start with a strand 1. And I promise I did not prepare any numbers. I totally picked the numbers out of the hat. You know? So we're going to have 4 different nucleotides. A, G, T, and C. You following me so far? Awesome. So let's say each strand has a total of 50 nucleotides. Okay? You with me? Let's say strand number 1 has 13 A's. Okay? Just 13 total A's, whatever position it is. How many T's strand number 2 is going to have? 13. Everybody understands that? Okay. Now, strand number 1 also has 16 G's. How many C's is going to be in the strand number 2? 16. Strand number 1, on top of that, has 10 T's. So how many A's in the strand number 2? And strand number 1, 29, 10, 39, has 11 C's. So how many G's? 11. Now, since we're talking about on the right, it's going to be 50 as well, right? So what's the total number of nucleotides? In 2 strands. Total. 100. How many A's total? 23. How many G's total? 27. T's? 23. And C's? 27. Does that make sense? Now, you have to understand, and this is, so why it is so important? If we know the percentage of one nucleotide, if we can run, I don't know, a chromatography, some analysis of DNA, and we can establish, okay, this DNA molecule has 30% G. So if the molecule, the double-stranded DNA, has 30% guanines, how many cytosines? 30% G, 30% C. How many percent left? Total. 40, and they will be split 20 and 20 between A and T. Does that make sense? Knowing one percentage of one nucleotide, you can figure out the rest. Does that make sense? Okay? So it was originally shown biochemically that this is how it works. And then when complementarity rule was suggested, it explained these ratios perfectly. Okay? Now you have to remember, this rule can be applied only to double-stranded DNA. Does that make sense? Are we clear? Single-stranded DNA, look at this. Imagine, forget about strand number 2. Imagine that you have only this molecule. Look at the ratios. There's no equality, they aren't equal. You see that? So, in double-stranded DNA, the percentage of A equals the percentage of T. And percentage of G equals the percentage of C. Everybody is on the same page? We can move on. DNA can be denatured. This is what I meant when I said unzipped. So this molecule of DNA, the double-stranded molecule, can be separated, strands can be separated by applying heat or increasing the pH of the solution. Why it is important? The isolated strands can be copied by the special enzymes. Say DNA polymerase can amplify such strand and make an extra copy. Where did we see, in which method of analysis, of microbial identification, we saw an approach that involved copying of a particular part of the DNA? PCR, yes, I'm talking about PCR. So when scientists discovered that DNA can be fairly easily denatured, and what's important, renatured back to the native state, it led, I forgot the name of the guy. I know he's a jerk. Yeah, he is like a big jerk. The guy who invented PCR, he wanted a patent for the method, like didn't want to share it with anyone. So that led that guy and another dude, I'm terrible with names, like two folks, to develop PCR. They got Nobel Prize for it. It's really a handy tool. Okay, so that's polymerase chain reaction. Does that make sense to you? Now what, again, I want to highlight, you can break them apart, you can collapse them down, okay? And this is what some cellular enzymes can do as well. There are enzymes, they can separate the strands during the processes of DNA replication or RNA synthesis. And then those strands will collapse back to their native state. Any questions before we proceed? Now, RNA. RNA is really an interesting molecule, as you remember, and we will repeat it again because that's a really important part of bacterial genetics. RNA is mainly a messenger. So it turns out, with some exceptions, and I'll mention those exceptions, RNA is, generally speaking, cellular RNA is generally speaking unstable. Now, there are differences between RNA and DNA molecules. Bless you. So this is a sugar found in the DNA. And this is hydrogen at the 2' carbon. This is the sugar called ribose found in the RNA. You can see that at the 2' carbon, there is now a hydroxyl group. There is now an oxygen. This makes the molecule of RNA less chemically stable. And that statement that I have there, makes molecule less chemically stable, is a bit of an understatement. If you isolate DNA from whatever sample you have, and you forget it on the bench, and you come next day, it's fine. You can store DNA up to a month in the fridge, several years at -20 Celsius, and as long as the freezer running at -80. -80 DNA is practically undestroyable. This is how we got the mammoth DNA. It's got frozen. Okay? Now, RNA, if you forget it overnight on the table, just throw it away. RNA is extremely unstable, and as you isolate it, it starts to break down, basically. It's very chemically unstable, which is really explainable. It's messenger. It's that photograph of the recipe that you have on your phone. You don't need to store it. Okay? Another difference between DNA and RNA is that in RNA, there is a uracil residue instead of thymine. So the pair is going to be not AT, like in the DNA, but AU. Okay? Generally speaking, cellular RNA is single-stranded molecule. It's a single strand right here. But DNA, I didn't correct that word again. DNA, sorry, RNA can form not intracellular, sorry, I overlooked it, intramolecular base pairs. So look at this image on the right. This is the molecule of RNA forming base pairs between two different fragments. Now, how that would look, let me go back to here and just draw something. Okay, so this is, imagine that you have a single-stranded RNA molecule. Now, these green fragments that I've got in that line are complementary to each other. Does that make sense? They can form pairs. So basically, this molecule will fold in the structure, that will look like this. You have pairs, okay? This is called hairpin loop structure. Does that make sense to you? So usually RNA molecule is heavily folded in the cell. Like in this image on the right, you can see just one of these hairpin loop structures where this part is the hairpin and this part is the loop. Okay. You all know that structure means function. All these hairpin loop structures play certain regulatory roles. They can be sites of the protein binding, or if they formed, proteins can fall off the RNA. So those hairpin loops are heavily involved in the regulation of RNA function. Okay. Now, what are the main types? I just want to finish this RNA thing, and we're gonna complete the class for today. But what are the main 4 types of RNA that I want you to know? One is mRNA. It's a messenger. It's the part of, gene expression process. The gene is transcribed. Gene in the DNA is transcribed into mRNA, which transfers the genetic information, okay, in order to make a protein. mRNA is the template for protein synthesis. Ribosomal RNA. As you can get from the name, it's the part, structural and functional part of the ribosome. It is actually an essential component. It's the enzyme, in this case. Ribosomal RNA serves as an enzyme. There are many ribosomal RNAs, different size. And one of them serves as the enzyme forming peptide bond. tRNA, or transport RNA, transports amino acids. So this is tRNA here showing leaving the ribosome. This is tRNA transporting an amino acid. I'm sorry, transporting an amino acid into the site of the protein synthesis. So far, does that make sense? Those 3 pretty straightforward. I want to mention one that I'm asking you about. I'm sorry, cold starts to get in me. So, and one that I just recently learned about myself. So, microRNA. We'll get back to the description, but basically, the idea is pretty simple, as far as I can tell. So, I mean, it's not simple at all. So imagine that this is a molecule of mRNA. Okay. Now here is the hairpin structure. Okay. Now it's poor. Anyway, so that's your hairpin. Okay. Now this is pre-mRNA. The protein, I believe it's called dicer. I think so. It cuts this out, and you, for some time, has this mature microRNA. It's double-stranded, which is interesting. It's short. It's about 22 nucleotides long, each strand. And the function of this molecule is to bind to a certain part of mRNA, okay, and target this mRNA for the destruction by so-called Argonaute protein. You don't need to know the names of the proteins. What I want you to understand is that this microRNA, really small, okay, it can attach to a message and direct this message to the destruction. It's like a tag saying destroy. Does that make sense? So microRNAs are really very important elements of the regulation of protein synthesis in the cell. In certain conditions, cell may start producing microRNA that will destroy a certain message. Does that make sense? So this protein will not be produced. Viruses utilize microRNAs to hijack cellular gene expression. Sometimes they use microRNAs to shut down cellular innate immune response. Humans try to use microRNAs as the drug, which is terribly hard, but there are approaches basically, if somebody has a genetic disorder, if we would be able to develop a drug, basically microRNA drug, that would enter the cells, target the defective message, and direct it to the destruction, that would be pretty neat, wouldn't it? There are a lot of obstacles. I think there is 1 or 2 drugs actually FDA approved now for some rare genetic disorders. I didn't look it up. Now, this is the 4 types that you need to know. But the last thing that I'm going to tell you that I learned, and that's what I love about microbiology, and that's what is terrible for me as a teacher. These lectures that I was doing like this year, like updating, they old. They already, you know, historically old. Last month, I think, there was a manuscript published in PNAS, one of Proceedings of National Academy of Sciences, showing that herpes viruses can form circular RNA molecules in the nuclei of the human cells, infected cells. And those circular RNAs are incredibly stable. I told you that RNA is unstable. This group, they managed to isolate circular RNA from the paraffinated tissue samples that were 30 years old. You can hardly get DNA from those. RNA was just fine. So we'll have to update this stuff, I guess. Okay. And that's not a complete list, but these 4 I want you to know. As I mentioned last time, we'll let you out early, so I will see you on Thursday.